All right, here we go. It's Monday, it's week 4. It's winter 2021. And here we go. So where have we been? We talked about fertilization, the event of fertilization, and we created our zygote, which is probably the beginning of life, I think it's safe to say. Then we took our zygote and we split it into two cells, but one zona pellucida. Remember, the zona pellucida is still, it doesn't normally split. If it splits, you're going to have twins. So all this splitting is going inside the zona pellucida. Yes. Are you kidding me? Oh my gosh. Yeah. Yeah, I think this happened in other classes last time as well. Well, don't know what to do. My internet is fine. So, I guess you guys can all go have breakfast. Or you can follow along on the slides if you want to do that. I know that's no fun, so I won't be taking attendance today then. I guess we got some of you on attendance, but so sorry about that. I'll just keep right on going, and you can email me your questions. And I will put in another complaint to IT. So, sorry about that. But, so we're on slide 2. And, yep. So, blastomeres. So we split our zygote into two blastomeres. And remember, those are cells. When cells split, you can call them daughter cells, but you really shouldn't call them daughter cells. They're called blastomeres. They're the same thing, though. And, yep, so the splitting is accomplished by mitosis. And, yep, and then we continue this process of splitting cells, which is called cleavage. So cleavage continues and continues, and somewhere between 16 and 32 cell stage, the embryo becomes known as a morula. Morula. All right, and now while this splitting is going, you also have to remember that we're moving down the fallopian tube. Usually fertilization occurs here in the ampulla, and then it just keeps getting squirted down this fallopian tube, and then it drops into the martini glass we'll talk about today. How does our little, I shouldn't call it an egg anymore, how does our little zygote or our little embryo, either one, embryo is probably better, how does it move down this tube? There's actually tiny cilia in the lining of this tube, and they're sweeping, kind of like the mucociliary escalator that you'll learn that sweeps junk out of your throat. Same kind of deal. So cilia are pushing the egg along, and if that ain't enough, we have peristaltic contraction going on, the same way that fecal material is kind of pushed through your intestines. So cilia and peristalsis is what pushes that along. First segregation occurs. So now we're going to start getting a little bit into molecular biology, but so some genes, the very first genes are going to get turned on. So late in day 4, the blastomeres of the morula begin to segregate into two groups. So our little cells within the zona pellucida, some of them start turning on different genes. And this turning on of genes, it creates two new cell populations, an inner cell mass cell population and a trophoblast. And this inner cell mass, that is you. You were once an inner cell mass. Trophoblast, this is all for support. So that protects you and nourishes you. Turns out placenta will come from that and such. So how does this work, this first segregation? Well, certain genes wake up and they start expressing, as genes will do, a gene product. via the central dogma. And I can't remember if I think we talked about that already. But specifically these gene products, they will tell the cells to do stuff. And specifically, they'll tell some of these cells to move toward and clump in the middle of our little embryo, and they will tell other cells to stay where they are, and basically they get pushed to the periphery of the embryo. We're on slide 6 now. And yep, and so this is the very first indications of embryonic cell expression. All right, and just here's the central dogma. I'm not going to go crazy because I know you all know this, but if you don't know this, I think you're expected to know this sort of stuff. Maybe Dr. Doe will go over it, or maybe physiology, they go over it as well. But central dogma, some include replication. I mean, you probably should include replication, but in interphase, the DNA is replicated, so you get a double-stranded DNA. And then the DNA is split apart, and it can be read by a creature called RNA polymerase. And remember, the genes are regions of nucleotides that contain information, really a blueprint on how to build stuff. So RNA polymerase reads this gene, and it creates a single-stranded RNA messenger called mRNA, which swims out of the nucleus, and it looks for a ribosome. That process, by the way, where RNA polymerase creates a copy of the message, an RNA copy, is called transcription. So the messenger RNA swims out of the nucleus, and it looks for a ribosome, and it binds to the ribosome, and the ribosome reads this message, this blueprint, and by inserting amino acids in accord with these codons, it creates the product, the gene product. And a lot of times it's a protein of some sort, but it can also be an MRI. It can be different things. But this is life right here. The central dogma is you're doing this right now inside your body. You're constantly doing this stuff. So that's kind of the central dogma. And go watch the Amoeba Sisters if you're lost on YouTube. Hopefully you're not lost. So the inner cell mass, we said that's you. That has an AKA. That darn Langman calls it the embryo blast. He's the only one who does that. So watch out for that AKA. That is one of the board books. It ultimately gives rise to the embryo proper, which is, we're going to go from this inner cell mass, as we'll see in this lecture, is going to form itself. It's going to segregate into 2 layers. But alternately, we're going to make a 3-layer disc, which becomes the human. And we'll get to that. The trophoblast, again, Langman calls it the outer cell mass, which I wish they wouldn't do. I wish they'd all get together, but nevertheless, you have to know that AKA as well. This is made of trophoblast cells. That's another concept. That trophoblast is, when I say trophoblast, I'm really talking about a collection of trophoblast cells. And this is going to give rise to the supporting tissue like the placenta. Tropho actually means nourishing. So here's what has happened. So we've had some genes turn on, and we're going to get into the weeds a little bit on this. We'll tell you what genes get turned on. But we had a random ball of cells, and now we have 2 populations of cells. We have our inner cell mass, and we have our outer layer, our trophoblast. And this is going to be this is you. The little human has started. Okay, whenever you see the little nerdy guy pop up, here it comes. So, and by the way, this stuff, this is for people who want to get a straight A in this class. Those of you competing to be the valedictorian. You need to pay attention to this stuff. And on boards, if you want to crush boards, this is for you. If you want to get by, maybe even get a B or so, you don't have to worry about this. Same with boards. These would be very advanced type questions, and I do put a lot of these videos on YouTube, and medical students have to know this stuff probably more than you guys do. But nevertheless, here it is for you, the story, for those of you who want to know everything. So for the blastomere, so those are the little cells, random cells in our little embryo. For them to move into the middle and clump in the middle to become the inner cell mass, we have to have two genes turned on. And those genes are called Oct4 and Nanog. So with those genes turned on, that's step one, to tell the cells to clump into the middle. And we'll see later these genes will be turned on again to help create hypoblast and epiblast. They're important for further development of the inner cell mass, I should say. So what do these do? So these two genes are going to turn on two other genes. Really, they're going to turn on a gene called Wnt, W-N-T, Wnt. And Wnt is the one who really turns on nodal. But the question could say, what Oct4 Nanog, what do? They turn on Wnt and nodal, but really Wnt turns on nodal, if you really want to get technical. So how do you become a trophoblast cell? What about the cells that stay to the outside? What's their story? So they have two genes turned on. So they have CDX2 and GATA3 turned on. Well, what do? They actually turn off the Oct4 and Nanog gene. So they keep the gene Oct4 and Nanog off. Remember, Oct4 and Nanog need to be turned on in order to turn on Wnt and nodal. So all four of these genes need to be on if you want to be you, part of you, the inner cell mass. So if you're going to be support tissue to the outside to prevent Oct4, Nanog, and really to prevent Wnt and nodal from turning on, you need to just turn on these two genes. So these are the genes that need to be on. They inhibit the production of Oct4 and Nanog. And therefore the cells don't migrate. They just stay to the outside, and they become the trophoblast layer, the trophoblast cells. Right? So we're on slide 12 now. Little rabbit hole, the uterus. We'll talk just a little bit about the uterus because, I mean, we're going to be implanting our little embryo right here. So we should know a little bit about this structure. So this is the uterus, and this is more gross 2 stuff. You'll get this more next quarter. But just in general, it has a fundus. It's kind of the roof of it, and it's in the anatomical position right now. So if a human female was looking at us and we're looking A to P at her, this is the picture you'd see. This is the body of the uterus, and the back is the body, and the front's been cut away. This is a coronal view of the uterus. And then this is the cervix right here. There would normally be some little kind of recesses right here. The vaginal cavity would kind of look like that. And these are called the fornices, or the fornices. Then we have os, an internal os and an external os. These are shut, and these have to open in order for sperm to get through. It's not just open normally like this. But in general, the uterus has these 3 parts. It has a fundus top, a body, and then the cervix is at the inferior portion of it. All right, histologically speaking, we need to talk a little bit about that, and I'm sure Dr. Dole will go over this more. But we have a perimetrium. Para means on the outside, so this is the outer layer. Not really exciting at all to us. It's a serous layer. It's also covered by visceral peritoneum, or peritoneum. I call it peritoneum because I did some medical school in a British type medical school, and they call it peritoneum, so it's stuck in my brain. But then we have, so this is the outer layer, it's pretty thin. Then we have a very thick muscular layer, which has a lot of smooth muscle in it. That's called the myometrium. It has a venous plexus and lymphatics. It grows like crazy during pregnancy. And then we come to our endometrium, which is more important to us because we have to implant our little zygote, if you will, or embryo into this layer. Here comes our zygote right here, this little blue thing. And it's made of 2 layers. So we have, confusingly, the stratum basale, which is right here. And why do I say confusingly? Well, if you've studied the skin yet, that's the magic layer that has the basal cells, which are stem cells. And this one too contains stem cells. So it's confusing because there's a stratum basale in the skin. That's the bottom layer of the epidermis. And now we have another stratum basale in the inner lining of the uterus. And then we have the stratum functionale. Now we don't have that layer in the skin, or you can call it the functional layer or the basal layer, or the AKs for that. All right, so the basal layer, again, that's where the stem cells are. It's outermost layer of the endometrium. If you're using the lumen of the uterine cavity, or the lumen of the uterus, the inside tube of the uterus, if that's the reference point, this is the deep layer of the endometrium. And this is the one that's going to give rise to a brand new stratum functionale layer. The stratum functionale layer is shed, so when females have their menstrual cycle, this is out of here. You lose that every month, but you don't lose the stratum basale where we can draw a basal cell here. Basal cells will rebuild this entire layer each month. Alright, so yep, not shed during menstruation. It also contains these important endometrial glands, or uterine glands. It doesn't have as many of them, but the deep ones live here. The stroma, which is the stuff between glands, kind of the gel that holds the glands and holds cells in place, is the stroma, is kind of the stuff. It's a lot more cellular, a lot more dense compared to the stratum functionale, which is loose, and that's why it can be shed so easily. Again, stratum functionale is loose. It's shed during menses, which is menstruation. It has surface epithelium, so it does, like all tubes have an epithelial lining of epithelial cells. This one is simple columnar, just like the whole GI tract, the esophagus, most of these, well, the end of the esophagus, the stomach, small intestines, large intestines, the rectum, is all lined by simple columnar cells. This one is also lined by simple columnar cells. Strangely, though, some of them have a cilia, kind of a sweeping cilia to sweep gunk out, to help sweep these unfertilized secondary oocytes that get shed and don't get fertilized. They could sweep it away. And then it has some important secretory cells, which are secreting some slippery substances, which will not allow unfertilized eggs to bind to the endometrium, because we don't want that. And this goes crazy, changes during menstruation. I'll let the others talk to you about that. And yep, and this is where most of the endometrial glands are. We're on slide 20. We're out of the rabbit hole. And here's a better picture of a uterus. Again, you can see the fundus, the body, and then the inferior portion is the cervix. This author correctly drew the fornices here. You'll see this next quarter. And you can see the internal os, the cervical canal. So the sperm has to get through this tight region. This is called the uterine cavity, or the lumen of the uterus. And then there's our fallopian tubes. Our little eggs probably right about here, heading this way. All right, martini glass. So about day 5, slide 21, day 5, the 36-cell morula drops into the uterine cavity kerplunk. So we've been traveling down the fallopian tube. There's actually a smooth muscle barrier that has to be relaxed in order for you to go kerplunk. So progesterone is the hormone that relaxes this little uterotubal junction, you can call it. And then you go kerplunk, and the uterine cavity is filled with fluid. It's not dry, so you literally fall into the fluid. And here's our little zygote, our embryo. Specifically, we could call it a morula because it's about 36 cell stage at this point. And yeah, so we're in the martini. And turns out that our little olive here is leaky, and it's going to actually take some of this sodium-rich fluid inside of it and develop a cavity. And so how can that be? How can our little tight ball of cells be leaky? Well, it turns out we've had a second kind of molecular biology event that has occurred. And we've created these really important little channels, which in physiology you're going to learn time and time again. We'll talk about them again in 5th quarter. But these are sodium-potassium ATPase pumps. So the genes that create these pumps have woke up, and now these first sodium-potassium ATPase pumps are being placed in the zona pellucida and in the plasma membrane, for that matter. So with these pumps working, you're taking in sodium, and the water follows. Sodium, well, water always follows sodium like a puppy dog. Chloride tends to follow sodium as well, like a puppy dog. Chloride always gets kind of left out of the discussion. But sodium and water are taken in. Yep. And so what? Who cares about that? Well, the water has to go somewhere, and it starts to form, kind of conjugate. Water is a very polar molecule, and it starts to clump together, and it starts to push apart, or push that clump of cells that we have in the middle. It pushes it upward, or up. It just pushes it to the side, and we start to get a new look of our little morula. And here's what I mean. So all the sodium and water has on slide 23, the sodium and water have created a cavity here, and that actually gets a nave. And our cells that were in the middle, our inner cell mass, got pushed to one side or another. And we'll just twist this so these will be up, because this is considered up. Where the inner cell mass pushes to, think of that as the North Pole. We're going to call that the North Pole. Well, we're not really, but the South Pole will be down here. But we got some direction to our little creature right now. Also, because this is a new structure, we have developed we've developed a cavity called the blastocyst, the blastocele, or the blastocyst cavity. The whole thing is no longer called the morula. It's called the blastocyst. Blastocysts. So say goodbye to the morula. When in doubt, you could always call it a zygote. When in doubt, you could always call it an embryo. But specifically speaking, we now have the blastocyst. Meet the blastocyst. And the blastocyst has a blastocyst cavity. Note that we still have a zona pellucida around this, but we're about to say goodbye to this zona pellucida. Right? The process of forming this cavity has a name. It's called cavitation. So cavitation. We'll see pretty soon this blastocyst cavity is going to get a name change. It's going to become a primary yolk sac, which in other creatures, not humans, it'll be filled with a yolk. Like you might be eating a yolk right now. You might be having an egg, which once started out as a blastocyst in chickens, and then converted it into a primary yolk sac, which morphs into a secondary yolk sac, and we'll get there in time. But so meet the blastocyst. We're still floating in the martini glass, though. We haven't attached to anything. Here's a beautiful electron microscopic picture of this. You can still see the zona pellucida hanging out here. The inner cell mass would be right here. That's you. And then the trophoblast cells make up the trophoblast layer. That's around the outside. And there's our blastocyst cavity. So we know this is not a morula. If I put a picture on the test and wanted you to name this, you certainly would not call this a morula, right? Because it's developed a blastocyst cavity. So this is the blastocyst. Okay, so we said that the inner cell mass, that's going to give some direction to this little ball of cells, and this will be called the North Pole. No, we can't do that. It would be silly. instead we're going to call this the embryonic pole. So the embryonic pole is kind of like the North Pole. And then the South Pole, directly 180 degrees away from the North Pole, we have the abembryonic pole, the AB embryonic pole, slide 25. All right. Yep, so that's the story with the poles. And here we can see a little bit more developed blastocyst. We also know from this picture it has not implanted, well, it obviously hasn't implanted because we have it here in a picture, but it still has the zona pellucida. So we can't implant into the epithelium of the uterus until we get rid of this. This will not implant. There's some receptors that the trophoblast cells express that need to bind to that epithelial cell. So we need to do something called hatching. So that's right. We have once hatched. So you guys have all hatched. So this happens about day 5. The blastocyst hatches from the zona pellucida. What that means is it says goodbye to the zona pellucida. And we're not exactly sure because there's so limited humans to study at this point in time. People who have abortions, they just throw all that stuff away and don't allow us to study, scientists to study that. So we don't know the full story, but some enzymes are obviously released. They think it's from the zona pellucida, like other animals, and I won't even get into them. But it burrows a hole in the zona pellucida. And here is an incredible electron microscopic picture of the hatching where you leave behind the zona pellucida. And again, we don't understand all the details of it, but it's if you don't hatch, you don't live. You can't bind, you can't become a viable fetus. So some hole is made by enzymes that we aren't 100% sure what they are, and then somehow the little blastocyst squeezes out of its zona pellucida prison, and now we have a naked blastocyst, no corona radiata. We lost that a long time ago, but no zona pellucida. It's just a naked blastocyst, and these are trophoblast cells exposed to the outside, the mean world, without any protection. Okay, so now that's floating around. Trophoblast, I kind of forgot to mention that. The corona radiata, slide 29, those cells were lost, degenerated away about day 2 while you're still in the embryo. So these don't get to go into the martini glass. Only the zona pellucida does. All right, so now we have another story with the inner cell mass. So some more segregation is going to occur. So the dorsally located epiblast, we're going to split the inner cell mass. It's going to split again into a dorsal layer and a ventral layer. So the dorsal layer would be in relationship to that embryonic pole, the North Pole. So we're going to make 2 layers. Right now we have a single layer inner cell mass. We're going to split it into 2 layers. These are very important words. You should memorize these. Epiblast and hypoblast. These epiblast cells, these are super stem cells. They give rise, every single cell in your body, you name it, has the ultimate cell it came from an epiblast cell. And it's going to go through some change. It's going to become an ectoderm cell. But these are magic right here. You could grow a new pancreas. You could grow a brain cell. You could, if we could work with these cells, they're absolutely amazing cells. All right, and then the hypoblast cells will be super important as well. Still some stem cells. They're going to morph into a whole bunch of stuff. But the epiblast cells are the key of life, because the epiblast will eventually kick the hypoblast cells out and replace them with a new layer, an endoderm layer it's called. But we'll get there. Okay, picture for you visual people. So here's what we have. We've got a new cluster that are up toward that embryonic pole. And we got another cell layer that's formed, the hypoblast. So epiblast and hypoblast. Blastocele is growing as well in our little blastocyst. So, oh gosh, here comes the little nerdy guy again. So hang on. So, because I know some of you want to go deeper. Epiblast cells. So what's the deal? How do you become an epiblast cell? Well, you have to have a gene turned on, and that gene is called fibroblast growth factor 4. And with fibroblast growth factor 4, it tells the cells to stay toward the North Pole, stay toward the embryonic pole. Don't do any migrating. Or if you, I mean, maybe this has fibroblast growth factor 4, then it'll migrate up here. So maybe this one doesn't. So this will migrate down here. So if you have FGF4 turned on, then you're going to migrate toward the North Pole, head north with this gene on. Okay, and then these cells that migrate toward the North Pole are called the magical epiblast cells. So how does F4, who turns on the F4 gene, or does it just wake up by itself? It looks like Nanog, which was the very first gene turned on. The Nanog gene and its gene product, Nanog, is what wakes up the FGF4. So it looks like kind of a chain reaction. But FGF4 has to be on. Okay. So how does the inner cell mass segregate? We're still working on the FGF4 from epiblast cells diffuse into adjacent cells. So here's kind of the story. What about the hypoblast? So FGF4 from these epiblast cells, which tell it to migrate toward the North Pole, it also gets out of the cells and it soaks into adjacent cells. So some of the cells get exposed to another cell's FGF4. Maybe the cell next door is a little slow at turning on FGF4. And if you're slow, the FGF4 will soak into the neighbor cells and stimulate the nucleus of that cell to secrete GATA6. So what does GATA6 do? GATA6 tells them not to migrate to the North Pole. Stay away from the North Pole. And so those cells are going to become hypoblast cells. So bottom line, to be a North Pole cell or a magical epiblast cell, FGF4 gets turned on. And to be a hypoblast cell, the GATA6 gene gets turned on. And that's the story with that. Pretty straightforward. So here's just another cartoon on 34. So FGF4, all the blue cells have FGF4 being turned on, and it's telling these cells to migrate up toward the North Pole, which of course is the embryonic pole, but you can remember North Pole right now. Later you'll remember embryonic pole. So the blue cells are heading this way, but some of these cells were slow to secrete FGF4, so the FGF4 that was secreted in the blue cells soaked into these cells and turned on GATA6, which prevented their own FGF4 genes from turning on. So these cells will get pushed down to the bottom. So that's the story with segregation of this inner cell mass. And we've created, that's you, that's the little human, but we've created now a bilaminar disc. This is called to be a bilaminar disc. So the little human is a bilaminar disc. And this is still the blastocyst. These are all blastocyst cells and blastocyst cavity. And yeah. All right, so yeah, meet the bilaminar disc. We got the bilaminar disc on 36. And great. We've also established an axis because we know what's up and what's down now. We know where the North Pole is. So it's said that the dorsal, that's the North Pole, ventral, that's the South Pole. We say the dorsal ventral axis is now there. So in electron microscopy, we can tell which way's up and which way's down. So yeah, epiblast cells are dorsal, hypoblast cells are ventral. Okay, just another picture on 37. Really already talked about that. I do like my pictures. That's why I have so many slides, because I like my pictures. Okay, let's do some more segregation. Another super important structure, super important. The little creature will die right now if this next step doesn't happen correctly. We need to make some new cells called the AVE cells, or the anterior visceral endoderm cells. So this is going to occur in the hypoblast cells. The cells toward the south pole are going to do something. So cells at the front end, or the anterior end, or the cranial end. Remember, I warned you that embryology is confusing because cranial and anterior aren't the same. But at this stage, a lot of embryology authors use these as AKAs. And that's because the cranial region is going to fold anteriorly. And some of the cranial structures stay cranial, but some of them move anterior. And they really should not do this because it confuses students and professors alike. But anyway, so our hypoblast cells, we're going to have some genes wake up in those cells, specifically the ones that are cranial. And they're going to create a special group of cells called the AVC, the anterior visceral endoderm cells. So what's the story here? Well, the nodal gene, which is turned off, or turned on, we've already talked about nodal gene is on. We need to turn that off. So if we turn off the nodal gene, it will create an AVE cell. So it's as simple as that. How does this happen is not so simple. How do you turn off nodal? So there's a bunch of genes. It's really important to turn off nodal in these cranial, these cranial located hypoblast cells. And so there's a bunch of mechanisms. So probably one of the most important genes that wakes up is called Dickoff1, or DDK1. And yeah, so that gets turned on in these future AVE cells. The DDK1 then blocks the expression of Wnt. And we said that Wnt is the one who turns on the nodal gene. So if you block Wnt, you're going to turn off nodal. And that's exactly what happens. So DDK1 blocks the Wnt gene, turns it off, and therefore without Wnt, the nodal gene will get turned off. Okay, and just in case you have a mutation in the Dickoff1 gene, there's some other genes that can help as well. And so we also have Lefty1 and Cerebrus1. Sounds like mobsters or gangsters or something. Lefty1 and Cerebrus1 are also turned on in these AV cells. So they help. So we have 3 genes and gene products that are designed to turn off nodal. Really turn off Wnt. And Wnt will, without Wnt, nodal can't be on. DDK1, that's like Al Capone. That's like the ringleader. And then we have Lefty1. That's his number 1 guy. And then Cerebrus1 is like the lower level mobster. But these 3 guys have to be on in order to shut that crazy nodal off. Okay? That's the story. So here's our story again. We've already created our bilaminar disc, but this is where DDK1 wakes up in these cells. And once this happens via the electron microscope, we can now not only know where up and down is, we know where front and back are. So we've established kind of 4 planes of the axis at this point. On slide 44 we are. Okay, so what are these cells? Why am I making such a big deal? Because they're really, really important. So we'll see later on that these AVE cells will release new genes will turn on within these AV cells that basically will make sure that the primitive streak, which is your nervous system, the first formation of your brain and spinal cord, starts with the primitive streak. And we don't want that forming cranially. The primitive streak actually starts caudally. And so we have to make sure that that doesn't form. And the AVE cells are critical. Horrible mutations can occur, and the fetus probably won't make it if this doesn't all work right. You can't have the primitive streak forming cranially. It has to form posteriorly. We'll talk about that primitive streak. You can believe that. Slide 45 now. So how do AV cells actually inhibit the streak? And we'll talk about this again when we talk about the primitive streak. But we have to secrete our nodal inhibitor signaling friends have to be turned on, or it's them that come to the rescue actually. So Cerebrus 1, the mobsters, Cerebrus 1, Lefty 1, DDK1, not only do they turn off nodal in themselves, they actually soak out of the AVE cells into the epiblast cells above, and they turn them. They diffuse, they soak into the cells above, and they turn them off as well. So it's pretty simple. And then they can't inhibit. I mean, they inhibit Wnt, and then without Wnt you can't express nodal. So it's the same process that turned the hypoblast cells into AVE cells, just extends to the cells above. So it's pretty simple, I think. What else? So there's more to the story. These AVO cells do something else. So they're super, super important for the formation of the heart, as we will learn. They're also super important for the formation of the prechordal plate. Who cares about the prechordal plate? Well, the prechordal plate, that's your forebrain. That's where your forebrain and part of the rest of your head come from. So these are very important cells, and they secrete a lot of stuff and orchestrate the formation of the heart, and indirectly formation of the head and forebrain. Okay, 47, 48 now, and we're 47, we're binding and implementation. So the presence of the developing blastocyst. We have to prepare the uterine. Let's back up. We have to prepare the uterus. Right now we dropped a morula into the martini glass, into the uterus. It was a leaky ship, started taking on water and developed a cavity. It's a blastocyst now. So now we got to get our blastocyst implanted into the uterus, into the uterine wall. So now how do we do that? So just the presence of the blastocyst in the uterine cavity, we don't know the whole mechanism, but somehow the uterus senses that there's a fertilized blastocyst floating around in there, and maybe it's hatched now so it can see the receptors. But we need to turn something off. Remember I said the epithelial lining, some of the cells secrete slippery stuff. That slippery stuff has a name. It's called mucin 1. And if mucin 1 is being secreted, it's too slippery to bind to our little blastocyst. So we need to turn that off. So it gets turned off. So that's good. So without mucin 1 going, we have a better chance of actually binding to the uterine wall. If you have too many mucin 1 genes and your uterine wall is too slippery, your fertilized embryo won't bind. So that's another problem with infertility, people who overexpress that gene. All right? But with it off, we can bind to the endometrium. We can dock and then get sucked inside the uterine wall. So binding actually occurs about day 6 or 7 with mucin 1 off. So usually the embryonic pole, the north pole, is the one that binds to the epithelial cells of the endometrium. Specifically, of course, there's some receptors. So the trophoblast cells of our little embryoblast, our little blastocyst, they express a receptor called L-selectins. We'll just leave it at that. And they're looking for their mate on the endothelial cells of the uterine lining of the epithelium. And that's called trophinins. And these two are looking for each other, and if they hook up, we have docking. We have successful docking. So here's our fertilized little blastocyst. There's our little human. That's you. And we can even see the AVE cells here. But there's an L-selectin, and it docks to the trophin. Again, if you have the trophoblast cells have to have genes turned on to make this L-selectin. Just like in the female, these epithelial cells here, columnar, they don't look columnar, do they? It's supposed to be columnar. But they have genes it's turned on that create this protein, this trophonin. And if you have mutations in those genes, here's yet another way that infertility can happen. So those receptors have to be expressed. If they are expressed and the slippery stuff is turned off, you're good to dock, and that's exactly how docking occurs. Okay, lots of stars. Guarantee this question is boards and for me and everybody. Slide 51, ectopic pregnancy. So usually, let's back up a slide, usually this docking occurs on the posterior body of the uterus. Remember, that's like the middle of the uterus, posterior body. And it's okay if it occurs in the front or the sides, but that's the only place it's supposed to occur. But this docking can occur in other parts of the uterus, and in the fallopian tube you can get docking, and even outside the body you can get docking. There's other mechanisms, these selectins and trophinins, there's other mechanisms for docking which is way too deep to get into, but this can potentially dock in other places. It usually doesn't, but it can. If it docks and binds somewhere else, that's called an ectopic. That means outside of the normal place. We'll talk about all sorts of ectopic gastric tissue, and there's all sorts of ectopia that occurs in the body, but it's called an ectopic pregnancy. And this can be life-threatening not only to the baby, but to the mom as well. It can create horrible pain. In fact, that's one of the causes of acute abdomen. You rush into the ER. Horrible pain. It turns out the docking occurred in the fallopian tube, and the fallopian tube is now ruptured. And so, yeah. So you should know where these common places are. The most common place of abnormal docking or ectopic pregnancy occurs in the ampullary region of the fallopian tube. So for whatever reason, the fertilized little zygote docked here and started to grow, and you can't have a baby like that. And then the next most common is the isthmic region of the fallopian tube. And then we have in the fimbria, these little fingers, the fimbrial region, it can occur. And then we start getting more rare. The ovarian, 0.5% of the time, the docking, the sperm actually came right out of the fallopian tube and fertilized the egg before it even got into the fimbriae. Fimbriae didn't grab it and suck it in there. So you can get a ovarian ectopic pregnancy. And then down in the cervix is another place that it should not be, 0.3% of the time. And then 0.2% of the time up here in the intestine, where the egg just got loose and the sperm hunted it down like hound dogs, and fertilization occurs. You can actually have a baby here. There have been cases where, as long as it's not in a little tube like that and there's room to grow, it can actually still be viable there. It's pretty rare. And then in this rectouterine pouch here, that's actually called the pouch of Douglas. Very interesting pouch. Well, you'll learn about. You can have the binding there as well. All right, let's meet a new cell type. I know you're ready. Everybody's excited to meet a new cell type. So the trophoblast cells, the ones that have docked, they start creating a new cell type, and this is it's almost like a cancerous cell type. These are called syncytiotrophoblast, these new weird cells. And yeah, they start out as cells. Let's draw some little cells on page 53 here. Drawing some little cells. Start out of these 4 little cells I'm drawing. And of course a cell has a plasma membrane, right? And what's weird is they lose the plasma membrane and they share a plasma membrane. So instead we have 4 little nuclei inside of one plasma membrane. Usually they don't bother to draw I'm just drawing the nuclear membrane here. That's a weird cell, right? So you have these clumps of these things all over. So these are called syncytiotrophoblast, and they're actually quite amazing cells. They're not cancerous at all, even though they behave. They're very aggressive. And slide 54 is everything I just said. They've lost their cell membrane, and they are invasive. They're the ones that grow into the endometrium and drag our little blastocyst with it deeper and deeper and deeper until finally it disappears and it's completely into the endometrium. So yeah, very cool. Here's a picture on slide 55 where the syncytiotrophoblast is invading. You can see that the trophoblast layer here in green, these are kind of pre-syncytiotrophoblast cells here because it should be one cell layer thick. But yeah, so this is growing and it's going to keep growing and getting bigger and bigger and bigger, and it's going to drag this epiblast right in, or this blastocyst right in. How does it push through the well, yeah, what's the secret? What do you mean it drags it? How do you drag through? I mean, here's the better drawing, right? There's columnar cells, although they look like pseudostratified. But how do you get through all these cells? And then you got cells in the matrix, and you got glands. How do you get through there? Well, you need some cutting mechanism. So we'll talk more specifically about these matrix metalloproteases in spinal anatomy. We'll get into the weeds a little bit on these. But for right now, let's just call them MMPs. So they secrete MMPs, which are ACEs. ACEs usually cut protein, and they just literally cut their way between the endothelial cells. And there's some MMPs are very powerful proteolytic enzymes, which can cause premature degenerative disc disease, as we'll see in spinal anatomy. But on slide, I guess this one's covered up, slide 57, that would be, in this cartoon, I drew some little dots here representing the MMPs, and that's how how our little blastocyst is going to cut its way in. Okay, slide 58. Everything I said, these syncytiotrophoblasts take over, and they get more and more and more, and they pull the blastocyst. And now the name of the game is, once we're out of the martini glass, we need to get some blood, because these cells, they're living cells, right? They need blood, so we need to hook up with a blood supply, which we'll talk about next time. But slide 59 here, you can see the syncytiotrophoblast has gotten way, way bigger. In fact, we have some cavities that have developed, which are going to become a source of blood. And the name of the game is the syncytiotrophoblast. Here's mom's blood vessel right here. Syncytiotrophoblast is going to drive right into mom's blood vessel and tap it, and the blood is going to start to accumulate in these cavities, which again we'll talk about next time. But that's kind of another part of the goal. So not only does the syncytiotrophoblast drag the blastocyst in, it also is looking for mom's blood vessels, and it taps into those to keep things alive. And if that's not enough, it does some other amazing things, these syncytiotrophoblast cells. One thing it does, as soon as it gets into mom's endometrium deep enough, genes turn on within the cytotrophoblast cells that secrete a very important hormone. I bet you've heard of this, HCG, human chorionic gonadotropin. That's one thing that steroid-using bodybuilders and cheating athletes have to actually take HCG to prevent the males do, to prevent the development of female breast tissue from developing. So that's another thing people don't know about bodybuilders. They have to take shots of HCG. But I mean, that's way beyond the scope of this. But so what does this stuff do? What's HCG do? Well, it's hormone, so it can travel all over the body. I don't know, do you know what hormones are? Hormones are molecules that get into the bloodstream and travel great lengths everywhere in the body, and they find a target receptor, bind to that receptor, and it turns on the central dogma to do something. It could be miles away. And in this case, the HCG goes back to our friend, the Corpus Luteum. Remember that lecture that you had to watch because we had a holiday? So I told you go watch, I think it was week 3 or week 2. Make sure you watch it. That was the most complicated lecture. So it's on YouTube. Make sure you watch it. I gave you a link in the description. because oogenesis is tough, a tough subject. And yeah, and that was ovulation genesis. But anyway, so HCG, what does it do? Turns out that the corpus luteum has receptors that will bind HCG, and it stimulates the corpus luteum to stay alive. Normally the corpus luteum, which was, this was the primary follicle, and it morphed into a secondary follicle. This was kind of what was left. Remember, the primary follicle houses the primary and secondary oocyte. The secondary oocyte is what got fertilized. I have a picture we'll look at here in a second. But normally it withers away and dies, but the HCG binds to it. It keeps it alive, and it keeps it. If it stays alive, it does its job, and one of its jobs is to secrete progesterone and estrogen. And it does so for 12 weeks until it finally dies. But by then, the placenta will have formed, and that will secrete huge volumes of progesterone and other hormones. Takes over. So the corpus luteum hands off its job responsibilities to the placenta after about 12 weeks. And recall the corpus luteum was the remnant of the ovarian follicle. Remember this picture. So this, I went over this on that YouTube lecture. But let's see how we're doing on time. Oh, we're kind of don't like to go much longer. But anyway, this is what we covered in that video. But we said that primary follicles woke up. You have a bunch of primordial follicles in your ovaries right now, or some of you do, and they're hanging out waiting. They may wait another 20 years before they wake up. But about 50 each month, about 50 primordial follicles wake up, and they start what I call the great race. And out of those 50, the dream, and even the ones that are in hibernation, the primordial means hibernating still. The dream of every primordial follicle is to be this guy right here. And that's a secondary oocyte. Of an ovulated secondary oocyte, for that to get sucked in the fallopian tube, and the ultimate dream is to get fertilized and create life. So out of these 50 racers here, only one of them normally will ovulate. Maybe you'll have twins, maybe two, but usually only one will ovulate. And we talked about all these stages, how the primary follicles got surrounded by granulosa cells, and then once a cavity developed in those granulosa cells, we got a name change to the secondary follicle, and then the secondary follicles have a chance to ovulate, so they're excited. And then during about day 14 of the menstrual cycle, you get a huge release of LSH and FSH by the female, and that stimulates one or two lucky ones to actually start the process of meiosis. Remember that these primary oocytes are frozen in prophase 1. But once they wake up, the lucky ones that wake up, they go through meiosis 1 and they start meiosis 2. They get stuck, unfortunately, in metaphase 2. But nevertheless, and look at how big this follicle, this is now called a graphene follicle. And the graphene follicle is the one that gets to shoot out the secondary oocyte. And remember, we said, if you watch the video, there's actually smooth muscle cells that have developed in this graphene follicle, and it helps squeeze and contract and shoot this out. It's actually an amazing process. But here, the point of the slide is, after this thing is out and away and down the fallopian tube, the remnants of that graphene follicle, they morph into this corpus luteum. And this is what has the receptors. I'm just drawing here on slide 62. This has a receptor from drawing a HCG molecule, binds to them and keeps it from withering away into nothing. All right, let's do a little more housekeeping and then we're out of here. So we've had a trophoblast layer for a long time. And this is just a house cleaning thing here. And I got a little shame. The authors are all over the place. I mean, once this term changes names, they should leave it, and it drives you crazy because they forget the name change and they go back and forth between trophoblast and cytotrophoblast. So once the blastocyst gets pulled into the uterus and it's not in the martini glass anymore, we don't call that outer layer the trophoblast layer anymore. Instead, those trophoblast cells and the trophoblast are gone. It's renamed the cytotrophoblast because now it's inside. It's a simple name change, but if you ever try to read the embryology books, which I have some students trying to do right now who are studying for boards, and they're emailing me like crazy, going, oh my God, this is such a mess, and the blue book that prepares you is errors, and it's just, embryology is just a mess. But just watch out for that if you're reading. Some authors forget the name change and they might go between. But these are the same. Trophoblast cells and cytotrophoblast, they're the same. It's just a name change. Right? And there's just another picture in 64. So, yep, cytotrophoblast. No more trophoblasts. Goodbye, trophoblast. So protection from mom. So another thing, so if we're inside of mom's tissue, why doesn't her immune system attack our little blastocyst? And that's a good question. Well, the cytotrophoblast cells, which gave rise to the syncytiotrophoblast cells, but cytotrophoblast cells, not the syncytiotrophoblast, cytotrophoblast cells release something called EPF, early pregnancy factor. And that is an immunosuppressant, and it protects the blastocyst. And it's kind of a localized effect. It doesn't turn off mom's defenses elsewhere, thank goodness, in the body. But it kind of calls off the anti-inflammatory effect. All right, and there's another mechanism we'll look at as well, but that's one. So another cavity, we'll do one more cavity and that's it. So right now we have that trophoblast cavity. But now let's meet another cavity. So, and this is as the syncytiotrophoblast is pulling deeper and deeper and growing. Another cavity forms between the cytotrophoblast and the epiblast layer. So this cavity is called the amniotic cavity. And that name should ring a bell because we've all heard of amniotic fluid. And this is a really important cavity. It will eventually completely surround the little human. So on slide 67, we can see a little bitty baby amniotic cavity has formed right here in the epiblast tissue. And yeah, there it is. And what happens next? And well, this is 68. This is where we're going. Look at how big this amniotic cavity has got. It completely surrounds the little human here. So we'll get to that. All right. So the amniotic cavity, a new layer of cells is going to surround it to kind of wall off it, because we're going to, and these are going to secrete amniotic fluid. So these are called amniotic or amnion cells. Some call them amnioblast. If you're in Europe listening to this, Bauer is a medical school book in Europe and some colleges. And amniogenic cells, they call them amnioblasts, but we call them amnion cells. And the amnion cells make up the amnion. So we have a new layer. And it's important. Some consider it the fourth extraembryonic membrane. And yep, it's so important that some call it an extraembryonic ectoderm. But we're not going to do that. That's Carlson. But I understand it's so important. There's 3 germ layers that are super important to development. This one is so important that some call this a 4th germ layer. But yeah, so these cells come from epiblast cells, give rise by mitosis, make this new layer. And the amnion cells surround the amniotic cavity. And really as simple as that. I'm just looking at a picture on 72. The amniotic cavity is much more developed now. The amnion layer and the amnion cells are right there. And yeah, so now we got our bilaminar disc. That's the human. And now we're going to start talking about forming a new layer walling off this blastocyst cavity. We're going to create the primary yolk sac in the next video. Okay, that is it. Sorry about the, 2 Mondays in a row. So I wonder if other professors are having the same trouble. I'll find out because I have a meeting right after this. But yeah, I'm sorry. No, annoying for both of us, for all of us. All right, see you guys later. I'll hang around for questions in case anybody just listened to the lecture live. Let me shut this down. I definitely don't want to lose this one.